Okay, this is photosynthesis, monitoring photosynthesis with carbon dioxide uptake. So make sure that you read the introduction before proceeding to the lab because it explains a lot of what's going to be going on in the lab and what you need to know. So first of all, you will need to answer the questions. What is your prediction for the experiment? So just read the options and choose the one that you think is correct. A, as photosynthesis progresses, the level of carbon dioxide will increase. So just think about, is carbon dioxide a participant or the product of this reaction? So if it's a participant, then it'll be consumed during the reaction and the amount will decrease. But if it's a product, during the reaction, the amount of product should increase. Okay? So let's think about it. If you can't remember, just go back and look at the um, photosynthesis. Oh, let me see. It kicked me out. So let me see. Hold on. Go to photosynthesis, carbon dioxide uptake. So look at the formula of the photosynthesis. Let's see what it is. Right here. 6 CO2 plus 12 H2O. Using the energy of light, we get oxygen as the byproduct, then water and glucose. Okay? So carbon dioxide is the reactant. So it will be used, okay? Consumed by a reaction. So the amount of carbon dioxide via photosynthesis should decrease. So when we make predictions about the experiment, we read, as photosynthesis progresses, the level of carbon dioxide will decrease. So yes, we think so because it's a reactant. All right, so we choose the option A. So what is your strategy? So let's read what we're going to be doing um, during the experiment. So how we're going to design our experiment. First option, I will place a plant in an environment with carbon dioxide and light for photosynthesis to occur. I will use pH indicator to monitor the level of carbon dioxide. I will compare the results to a negative control with a plant. Okay? The next option, I will place a plant in an environment without carbon dioxide and in the light for photosynthesis to occur. I will use a pH indicator to monitor the levels of carbon dioxide. I will compare the result to a negative control without a plant. So, do you think that we do need carbon dioxide? Of course we do, because it is needed for photosynthesis. So plants absorb um, carbon dioxide um, to be able to produce glucose and oxygen. Okay, so option B is obviously wrong because without carbon dioxide, photosynthesis will not occur. And the last option, C, I will place a plant in an environment with carbon dioxide and light for photosynthesis to occur. I will use a pH indicator to monitor the level of carbon dioxide. I will compare the results to a negative control without a pH indicator. So that's also wrong because without a pH indicator, we won't be able to see if the acidity or basicity of the solution has changed. Okay. Um, if you need to go back and see what the pH indicator is going to be, what color is going to be in acidic solution when carbon dioxide is um, dissolved in water, and then when the amount of carbon dioxide decreases, what color the indicator will be, how it's going to change when the pH of the solution changes. Okay, so we choose option A here, okay? Because we do need carbon dioxide for photosynthesis, and we need a pH indicator to monitor the level of carbon dioxide, okay? And we do a negative control with the plant. So just moving to phase 2.